Now, one of the main issues here is for any linear DNA in eukaryotic chromosome, okay, we have the usual replication, but always remember the process cannot complete at the 5' end of the daughter DNA strand. We can't do this. Okay, so because our DNA polymerase only can add nucleotide to 3' ends, even we have the Okazaki fragments, when the primer is removed, it cannot be replaced. With the DNA. So therefore, there's no 3' available for nucleotide addition. So at the end of the DNA replication, we have small portions left unreplicated and a small portion single-stranded segments of the DNA lost with each cycle. So it means that basically every DNA replication, our DNA actually become shorter and shorter. So as a result, repeated round replication produce shorter and shorter DNA molecule with uneven end known as a stagger end, and eventually loss of the vital gene would result in the cell death. Okay, so let's look at why the DNA replication will cause this. Okay, in the linear DNA is not complete. Now let us look at DNA replication in general. These are the replications. Okay, so now first we do have our DNA. Okay, DNA. Okay, so this is a 3' end. This 5' end of the parental strand open up. 3' end, 5'. Okay, so what we normally do here is at the 3' end we attach with the primer. This is RNA primer. RNA primer start with 5'. Correct. The primase actually adds the RNA primer. So next, because of the double-stranded regions here, then DNA polymerase now adds the nucleotide-free activated DNA nucleotide one by one all the way. So the direction, can you see that start with 5' all the way end with 3' ends? Can you see that? Okay. Now, the directions is this way. Why? Because here is 5', here is 3', so always from 5' to 3', no problem. So DNA replications complete now. Now, how about at the lagging here? Same thing, we add the primer. When I add the primer, this primer start with 5'. Can you see that? So the DNA polymerase now recognize the double-stranded regions here. And next, can you see that? So this is 3'. 5', 3'. Correct. Okay, so directions, this direction. Why? Because can you see that primer start with 5' and with 3'. Okay, now what actually happened now? Because our DNA cannot be mixed with RNA and also the DNA, so therefore this RNA primer will be removed. Can you see that? This RNA primer will be removed later. When it is removed, then you can see that what actually happened. Now, DNA polymerase want to add. This is a missing part. Can you see that? Missing. Okay, the two missing parts. So if our DNA polymerase want to add, the direction will be this way, correct? So if this way become 3' to 5', if I want to add this way, 3' to 5', which is impossible to be done. Okay, so what is the consequence here? So every time of the DNA replication, our DNA become shorter. Are you clear? So DNA becomes shorter because the RNA replication is incomplete. So over a period of time, you can see that our DNA becomes shorter and shorter until the gene actually eroded. So what actually happened now, we can see that this is our DNA. We have the vital gene. So this is a gene, let's say code for the cell survival. Okay, another gene code for cell survival. Okay, so let me. Now, what will happen now? Look at this, look at this. What will happen? Okay, every time DNA replication, part of it cannot be replicated. The 2n, 1n, 2n. Okay, so now what happened? The second round. So can you see that the gene? What happened here? Original gene. Can I see that? So in this case, we can say that gene loss take place, or we say that it's gene erosion take place. Means that now the gene is incomplete, so means that after replication, the cell actually can't carry out the function, cell die. Okay, the cell will die. Okay, so basically what actually happened here? Okay, so from one cell, mitosis take place, become two cell. Okay, no problem. Okay, now if the cell continue to carry out the mitosis, for example, because of the gene erosion, the cells can't survive. Because of what? The vital gene, missing of the vital gene. So there's a gene erosion take place. And this gene erosions actually cause the cell death. So when cell death, so no mitosis, so can you see that the organs cannot replace the cell? Anything cell die, the organ cannot. So it would death, the cell death will actually eventually cause the aging process. One of the example, organ failure. Means that we can't replace the number. Let's just try to imagine we have 1,000 cells in one organ. Now the cell keep on dying, but we cannot. Right? We can't actually put back the numbers to replace the loss of the cells. So therefore, organ failure actually take place. Can you see that? Okay. So you can take a photo here if you need. Then we look at how we have this protective mechanism called telomere, right? So at the end of the DNA, always where the small part is left unreplicated. And you try to imagine the unreplicated one will continue to be unreplicated again in the second round of the DNA replication because they act as the template until they have the loss of the vital gene. Vital gene means important gene. Okay, that would cause the cell death. Okay, and then this cell death we cannot replace, right? We cannot replace, so therefore, organ failure take place. So always remember, in the growth process, okay, in the growth process, so if you look at this, when we are young, the rate of mitosis, can put this way. Okay, when we are young, so the mitosis rate more than cell death. Cannot. So therefore, you can see that we grow. Okay, we become taller, organ become larger. We try to imagine that if the organ then develop, okay, the organ becomes so small, cannot be continue to grow. So therefore, you can see that because of the mitosis more than the cell death, okay, the rate. But when it comes to adult, you can see that the rate of mitosis relatively equal to the cell death. So therefore, you don't have the growth anymore. Can you see that? Because okay, two cell die and then we replace the cell, okay, and so on. Okay, so that is why we don't actually continue to grow taller and the organ cannot continue to grow bigger. If the mitosis rate at this moment is higher than the cell death, what will happen? Cancer cell may develop. Okay, when we are old, what happened here is the mitosis rate less than the cell death. So it means that more cell will die and mitosis can't continue because what? The cell die already because of the loss of the vital gene. So therefore, organ failure start to take place. Okay, a lot of times, why organ failure, heart cannot functions well, and then Alzheimer, loss of the memory because of the cell die. Okay, the death of the cell. But this thing won't happen when we have the circular DNA because there's no end, so the shortening of DNA does not occur. So students always confused why circular DNA, they don't have these issues. Let me show you guys. Okay. So look at the prokaryotic cell. Okay, prokaryotic DNA replication. So because the DNA is circular DNA, look at this, circular DNA. Okay, double-stranded. Okay, double-stranded. So what will happen here is, so during DNA, they will act as a templates. Okay, so separate two molecule. Okay, two molecule. So if this part is the origin of replication, okay, origin of replication, so you have the primer. Okay, you have the primer here. Okay, so primer start with 5'. Can you see that? And then DNA polymerase is going to continue to add the DNA nucleotide one by one all the way until here. Okay, so even now, try to imagine, okay, now, if let's say the removal take place, we remove this primer. When we remove the primer, we realize that this side start at 5', this side 3'. Can you see that? So DNA polymerase still can continue to add, and then ligase seal the sugar phosphate backward. So it means that no loss of DNA. Can you see that? So another side 3' here. Okay, so start with 5'. So DNA polymerase will continue to add DNA nucleotide all the way, free activated DNA nucleotide all the way until here. Okay, then no problem. When you remove, DNA polymerase continue to add. Can you see that? Directions, right? It's 5' to 3' end. So therefore continue to add. So continue to add, and ligase seal the sugar phosphate backbone, therefore forming the two molecule of the circular DNA. Okay, so the basic principle is here, but in the exam, so question asks why there is no gene loss in prokaryotic cell. Then the answer very simple, because their DNA is a circular DNA. When a circular DNA, they don't have the open end. When they don't have the open end, so therefore the DNA replication is complete. So when it's complete, so therefore there's no gene loss. Okay, so I can take a photo. So it means that the primer is removed, but there's no gene loss. Yeah. Okay. So I off the record first. So you look at this diagram. Okay. So you can see that first, this primer, red color primer. When primer is removed here, no problem. We can continue. Can you see that? Because 3' end is available. But now you can see no 3' end. Can you see it? When we remove the primer, we can continue because 3' end is available. But now no 3' end, we only have 5'. So if you want to go through this direction, it's impossible because it becomes 3' to 5'. So impossible, right? Mandatory. So every time now this becomes template, becomes shorter and shorter, the shortening of the DNA molecule actually take place. Okay. So how the protective mechanism we have to do in this case? So every eukaryotic chromosome DNA, they have the special nucleotide sequence called telomere. Highlight this telomere. So this telomere, okay, will be formed at the end. So what is telomere? I highlight telomere, they are non-coding. Means that they are not code for any protein. Their guanine-rich DNA sequence consists of repetitions of short DNA nucleotides. So in human, TTA, GGG. Okay, so look at this. Okay, what actually happened here is, and we're going to compare. Okay, we're going to compare between these two scenarios. Yeah. Okay, first scenario, without, okay, without a telomere. So what will happen? You see, without telomere, this is the DNA. Okay, so this is the vital gene where the gene is located. Okay, and drop. Where is the gene located? Yeah. So those are the vital gene. Gene number 1, gene number 2, 3, 4, and 5. Okay, and we do know that every round of DNA replication, try to imagine, so first round of DNA replication, DNA becomes shorter. Okay, no problem. Vital gene is still there. Second round of DNA replication becomes shorter. So at the second round, can you see that this part? Oh, sorry. Can you see that the vital gene lost already? Can you see that? Gene erosion. Okay, after, for example, 2 rounds of replication. Can you see that? This is without telomere. So with telomere, what it means? Now, look at this. With telomere. So what's telomere? If you look at the DNA now, same length of DNA. Okay, so we have gene 1. Okay, we have gene number 2. Gene number 3, same length. Gene number 4. Gene number 5. Okay, gene 1, vital gene 1, 2, 3, 4, 5. Now, what actually happened here is with telomere. Telomere actually is the repeating sequence. Okay, repeating sequence. TTA, GGG. They are non-coding, so repeat how many times also won't affect. It's not a mutation in this case, but basically their function is to lengthen. There's a telomere, lengthens our DNA. Okay, made longer. Can you see that? Okay, lengthen the DNA. So this is where the telomere is found. Can you see that? This is where the telomere is found. So what happened now? Look at this. Yeah, look at this. Sorry, this white color here. Now look at this. So first round on DNA replication, shorter. Second round, shorter. Third round, number 4, number 5, number 6, number 7, number 8. Can you see that? Only up to number 8, then you can see that gene loss takes place. So basically, what is the function of telomere? Telomere actually make your DNA longer and they delay gene erosion. Can you see that? Not so soon. Okay, without telomere, after 2 rounds, with after 8 rounds. So means that we can have more mitosis and cell division to take place. Can you see that? So therefore, we say that telomere delay the gene erosions. Okay. Now, but here we have a bit of confusion that we need to actually clarify here. Okay, you can take a photo of this, and then I will erase this part and tell you more about, okay, the gene erosion. So it has been proposed that shortening of the telomere is somehow connected to the aging process and also of the certain tissues, even aging of organism as a whole. So first always is like this, okay, cell death cannot be replaced, and then start with the organ failure and continue with the what we call this, the aging and death. Right? So that's why we have a limited lifespan. You cannot continue to grow and grow and grow unlimitedly. If you grow and grow and grow unlimitedly, that is either cancer or stem cell. These two types of cells, they can continue to grow and continue to multiply, continue to divide unlimitedly. So we actually can restore the original length of the telomere actually by having the enzyme called telomerase. So telomerase catalyzes the lengthening of the telomere in eukaryotic stem cells. Highlight stem cell, not all the cells, but not the somatic cells. Because telomerase activity is low in most of the human somatic cells, but the activity varies from tissue to tissues. Like liver cell, because they can regenerate, so the telomerase activity is slightly higher when compared to other, like for example, our kidney, our heart, where you won't be able to regenerate that. So restoring the original length. So active telomerase in stem cell result in the telomere of maximum length in the zygote. So if you look at this diagram, if you look at the telomerase, the structure of telomerase is enzyme. It's the enzyme with a RNA template. So we know, right? TTA GGG is our telomere. So in this telomere, you can see that this telomerase carry the RNA template that can have the pairing. Can you see that? GNC, TNA pair. So leaving the empty gap here. So the empty gap act as a template so that the new DNA can be added. And then they'll continue and continue to make this telomere become longer. Later, I'll show you the video clips, then you'll know. But one thing I want you guys to change here, this is not called telomerase, it's telomere. Sorry, my mistake. So I'm going to show you guys the process, but just for your information only. So don't need to memorize, or remember the process, how telomerase actually functions. So when you use the word reverse transcriptase, means that this enzyme use RNA as a template, to make DNA. So we're going to learn about reverse transcriptions and process in A2 also. And this enzyme also have a special what called telomerase RNA act as the template. And also some protein complexes. So TRT or TERT actually is a reverse transcriptase. It's an enzyme that uses RNA as templates to make DNA, which is a class of enzyme that creates single-stranded DNA using single-stranded RNA as a template. So by using this TRC or TR, so this enzyme can add 6 nucleotide repeating sequence TTA-GGG to the 3' ends of the DNA. So these TTA-GGG repeats are called telomeres. So the template regions of TERT is 3', so don't need to remember, guys. Do not need to remember. So telomerase can bind to the first few nucleotides of the templates to the last telomere sequence on the DNA, add new telomere repeat sequence, let go, realign. And the main important here is telomerase reverse the telomere shortening after each round of DNA replications. So with this, I've done the telomere. We're going to continue with the stem cells later. So I'm off the recording now.